Welcome to The Explainer. Today, we are decoding a major scientific breakthrough in Alzheimer's research, and it's all about a genetic mystery that has had scientists scratching their heads for years.
So for a long time, scientists have known about a specific gene that seriously increases the risk of developing late-onset Alzheimer's. They knew it was important, but they had no idea why.
You've probably heard of the APOE gene, right? Well, right after that, variations in a gene called BIN1 are the second biggest genetic risk factor for the disease. It's a huge player.
And that is the million-dollar question in genetics. I mean, knowing a gene is risky is one thing, but to find a cure, you have to understand the nitty-gritty mechanism. What's it actually doing inside our cells to cause all this trouble? That's the real puzzle.
Okay, so let's dive right into this mystery. We're going to follow the clues from a brand new study that has finally given us a clear and pretty elegant answer.
To really get what's going on, we have to shrink ourselves down, way down, into the microscopic world of our brain cells, and we're going to pay a visit to their essential, always-on-duty cleanup crew.
This whole process is called autophagy. You can think of it as the cell's built-in recycling and garbage disposal system. And it is absolutely crucial for keeping our cells healthy, especially our brain cells, which have to last a lifetime.
So autophagy isn't just one thing. It's this highly coordinated four-step dance. First, you capture the junk in a membrane. Second, you seal that membrane up tight like zipping a trash bag. Third, the full bag gets snipped free. And finally, it's hauled off to be recycled. Every single step has to happen in perfect order for any of it to work.
All right. Now let's zoom in on the two most critical steps here, sealing the bag and then releasing it. What's so cool is that these two jobs are controlled by two totally different teams of proteins.
On one side, you've got a crew of proteins called the ESCRT complex. They're the sealers. Their job is to zip that trash bag shut. Then on the other side, you've got a protein called DNM2. It's the cutter, and its only job is to snip the sealed bag free so it can get recycled.
Okay, so you can see the problem, right? This is a massive coordination issue. If you cut that bag loose before it's sealed, well, that's a cellular disaster. You'd spill toxic waste everywhere. The timing has to be perfect.
And this is where our mystery protein, BIN1, finally makes its grand entrance. And it turns out, it's not the villain we might have thought it was. No, it's the master coordinator.
This new research revealed BIN1's true job. It's a switch. It's like the middle manager on the factory floor that connects the sealer and the cutter to make sure they work in perfect harmony.
Here's how it works. It's actually pretty simple. As long as that trash bag is still being filled and sealed, the SCRT complex, the sealer, is literally holding on to the BIN1 protein. It's got it in a tight grip.
And get this, while BIN1 is being held by the sealer, it's actively putting the brakes on the cutter, DNM2. It's basically shouting, "Whoa, hold on, not yet." It stops it from cutting too early.
Now, when the sealing job is finally done, the SCRT complex changes shape, and it just lets go of BIN1.
And the second BIN1 is free, it stops putting the brakes on DNM2. The coast is clear. The cutter is activated, and it swoops in to do its job. Snip the perfectly sealed bag free. It is just a beautiful, elegant biological switch.
So that's how it's supposed to work. The big problem, the whole reason we're talking about this, is that this elegant system is exactly what breaks down in people with that Alzheimer's risk gene.
And here's the real kicker. The genetic risk isn't because the BIN1 protein is broken or mutated. Nope. The problem is that the brain's immune cells, these things called microglia, just start producing way too much of this perfectly good protein. And the result is a total disaster for the cell. With too much of this coordinated protein floating around, you get a massive pileup of these uncleared trash bags. It's a complete traffic jam in the cell's garbage disposal system.
You can think of it like this. The switch gets stuck. With all that extra BIN1 around, the brakes are just always on. The cutter protein, DNM2, is constantly being held back, and the whole recycling pathway just grinds to a halt.
Okay, so we've got a cellular traffic jam.
But how does that actually connect to the devastating symptoms of Alzheimer's? Let's connect the dots.
Well, when the brain's immune cells, these microglia, can't even clean up their own mess, their other jobs start to fail. They can't clear out those toxic amyloid plaques we always hear about. They get stuck in a state of chronic inflammation, which is terrible for the brain. They even help spread other toxic proteins like tau. Essentially, they get old and worn out way ahead of schedule.
Look, this discovery is a huge step forward. We've gone from just a vague genetic mystery to a specific, targetable mechanism. By understanding exactly how this little switch works and how it gets stuck, we've opened up a whole new and really exciting avenue for developing therapies to fight this disease.